Morning, it's a Dr. Ben update. So we'll look through a couple of papers this morning. I got them open in a couple of tabs, so I'm just going to click through them. What has come out over the past 2 days in bioRxiv, you ask? Yeah, and so here are the highlights that I've seen anyway. Um, Number one is that there is another study. So this is now a third study, and it's this technique called single nucleus profiling, where they're pulling out one cell at a time and looking at what it is making, what sort of genes it's expressing at the moment. And they can then tell what kind of cell it is and also what it's doing. And they can look for differences in age, and they can look for differences between males and females. And what they're finding are differences in age in males and females. So there's a little difference in the main receptor, the ACE2 receptor. So that goes up a little bit with age and gets a little bit broader. There are more cells that make a bit of it. The real difference, once again, for the third paper in a row, seems to be TMPRSS2. Remember, that's the second protein that you need in order to actually get the coronavirus into cells. It's the one that's going to actually sort of clip the spike protein and activate it so it can actually work. Basically take the safety mechanism off the spike protein. And so, that seems to increase in expression. Like there are more cells that express it, and they each express more of it as you get older. And so, we're making more of the things that help coronavirus get into our bodies as we get older. And so that's why we're getting more incidents of infection, like that actually takes hold in people who are older. Like I was saying, I think kids are probably just about as exposed as adults in terms of who has, run into the virus. But I think that the virus has a harder time actually getting into kids. Now, once it gets into kids, it seems to grow very well. They seem to have very high amounts of virus inside their lungs. And so it's once they're infected, they're probably very good at infecting other people, just as good as anybody else. So that's a nice message of equality right there. Yeah, kids can infect people too. We think, just based on the amount of virus they have inside of them. All right, so that's the first paper, and let's look at a second paper. And so the second paper is looking at the different kinds of cell expression changes, like what cells are doing. That's what expression means. It's like they have this library of maybe 25,000 genes and a bunch of variants of each of those that they could be making at any one time. And the particular set that they're making kind of tells you that, oh, this is a neuron in the brain, this is a hepatocyte in the liver. They just make different selections out of the DNA, but they all have the same DNA that they're reading from. And so this one looks at the immune response, and what they find is that it's kind of a mix of good and bad things. They find that when the virus is inside of a person's lungs, or rather inside of these lung cultures that they make that come from somebody's lungs, but you remake it inside the lab, so you're not dealing with an actual person who's sick. And they find that those cells are making some interferon type 1 and 3. So like alpha beta interferon and lambda interferon for immunology types out there. You know who you are. Actually, you don't even need that, immunology types. You already knew that. I can't teach you anything. And we also know that these are things that are effective against the virus. And so part of the body's immune response does seem to be effective. At least it's made by certain cells. Cells that are the actual lung cells that are not macrophages. So they seem to be kind of doing the right thing. They also found that there is a lot of interleukin 6 that is made by these cells, and this is one of the messages that we think is responsible for how sick you get. Like the more interleukin 6 you make, the more sick you're going to get, just like that. So that's explaining why the cytokine storm is happening, and this does suggest if we can find more ways of potentially slowing down interleukin 6 production without actually shutting off any component of the immune system completely, then theoretically, we should be able to at least protect people from the cytokine storm to some extent and give their immune system time to figure out the answer to fighting the virus and then, go fight the thing. Once again, it's another study that says that interleukin 1, which is one of these other sort of classical molecules that really drives very strong immune responses, doesn't seem to be particularly involved in COVID 19. So there we go. Interesting. And yeah, this suggests that some of the approaches that people are taking with the IL-6 blockers and also trying to hold down the amount of virus, which is causing these particular changes that then set off the cytokine storm, it seems like those are the right approaches. And yeah, we're doing the best we can, like as a, I don't know, as a world, as a scientific community. Yeah. So they're good ideas. Nothing new is popping up. We don't have any new Achilles heels, if you like, to go hunting in this virus. But there actually are a lot of things that we know the virus needs, and that we can potentially take away with little drugs or different kinds of treatments. So there are a lot of clever chemists out there that are coming up things right now. I know. Yeah, I've talked to some of these, and it's going to be really interesting to see if they can come up with some new solutions for this virus. So thank you very much. That's our Dr. Ben update.